Role of embryo culture and the laboratory environment in optimizing RT success. In assisted reproductive technology, ART, the embryo culture process and the laboratory environment play crucial roles in ensuring the success of treatments like in vitro fertilization, IVF. The goal of RT is to create a healthy embryo outside the body and then transfer it to the uterus to establish a pregnancy. To achieve this, both the environment in which the embryos are cultured and the conditions provided during their development must be carefully controlled. First, embryo culture in ART. Embryo culture refers to the process of growing fertilized eggs, embryos, in a laboratory setting after egg retrieval and fertilization before selecting the best embryos for transfer into the uterus. The conditions in the culture environment are designed to mimic the natural conditions that the embryo would experience in the female reproductive tract. Successful embryo culture is essential for selecting high-quality embryos that are likely to implant and develop into a healthy pregnancy. Stages of embryo culture: fertilization and initial culture. After egg retrieval and fertilization, either through traditional IVF or ICSI, the embryos are cultured in a nutrient-rich medium that supports their early development. The initial stages of embryo development, such as the 2-cell and 4-cell stages, typically occur within the first 24 hours. ICSI is often used when male infertility is a concern, allowing the direct injection of sperm into the egg to ensure fertilization. Cleavage and blastocyst formation. Embryos continue to divide and develop through the cleavage stage, where the embryo splits into multiple cells. The blastocyst stage, typically around days 5 or 6 of development, is considered the most advanced and is the optimal time for embryo transfer. During this stage, embryos form a fluid-filled cavity, and the inner cell mass differentiates into tissues that will become the fetus. Embryo grading. During culture, embryos are assessed for their quality and development. Embryo grading involves evaluating the appearance, shape, and rate of division of the cells. Embryos with consistent and organized development are graded higher and are more likely to implant successfully after transfer. Embryo selection. Not all embryos are viable for implantation. Embryo culture allows for the selection of the healthiest embryos, increasing the chances of success. In cases of pre-implantation genetic testing, PGT, embryos are also tested for genetic diseases or chromosomal abnormalities before being selected for transfer. Factors influencing embryo culture. Success. Culture media. The composition of the culture medium is essential for embryo development. The medium contains amino acids, vitamins, minerals, and growth factors that support embryo metabolism and cell division. The medium must also be designed to maintain the proper pH, osmolarity, and oxygen levels to ensure optimal conditions for the developing embryo. Incubator conditions. Embryos are cultured in incubators that simulate the human reproductive environment. These incubators are controlled for temperature, humidity, and gas composition. The temperature is usually maintained at 37 degrees Celsius, body temperature, and oxygen levels are carefully controlled to avoid stress on the embryos. Oxygen tension. Oxygen levels in the embryo. Culture medium are carefully controlled. Low oxygen tension, hypoxia, is typically used to mimic the in vivo environment, as it is believed that embryos develop better in lower oxygen conditions. Embryo handling. Handling embryos with care is critical, as embryos are highly sensitive to mechanical stress. Advanced equipment and sterile techniques are used to minimize exposure to contaminants or temperature fluctuations. 2. The laboratory environment in ART. The laboratory environment in ART is not just about the technical aspects of embryo culture, but also the overall atmosphere in which the work takes place. Creating a controlled, sterile, and optimal environment is critical for ensuring embryo development and improving ART outcomes. Key aspects of the laboratory environment: sterility. Embryo culture and handling require a sterile environment to prevent contamination. Contaminants such as bacteria, fungi, or viruses can severely affect embryo health and implantation rates. Clean rooms and specialized laboratory equipment help maintain sterility during all stages of the ART process. Controlled temperature and humidity. The laboratory environment must be kept at a constant temperature and humidity level to ensure that the embryos remain in conditions similar to the natural reproductive tract. Incubators, laminar flow hoods, and air filtration systems are used to maintain a consistent environment and minimize the risk of environmental stress on the embryos. Staff expertise. Skilled embryologists are crucial to the success of embryo culture and handling. Embryologists are responsible for monitoring embryos, performing biopsies for genetic testing, and selecting embryos for transfer. Their experience in handling delicate embryos is key to minimizing risks during the culture process. Environmental control systems. The laboratory environment should be equipped with systems that monitor and regulate gas composition, e.g., oxygen and carbon dioxide levels, temperature, and pH to ensure consistent conditions during embryo development. Embryo surveillance. Embryos are monitored regularly during the culture period to assess their development and select the most viable embryos for transfer. Advanced imaging techniques such as time-lapse microscopy allow for continuous observation of embryo growth without disturbing the culture environment. 3. Optimizing RT success. Through embryo culture and laboratory conditions. To optimize ART success, a few factors are critical. Optimization of embryo culture conditions. By maintaining ideal temperature, gas composition, and pH levels, as well as using the right culture media, embryo culture conditions can be fine-tuned to encourage healthy development and improve implantation success. Advanced monitoring and screening techniques. The use of time-lapse imaging and embryo genetic screening such as PGTA, PGTM, or PGTSR helps improve the selection of the most viable embryos for transfer. Time-lapse imaging allows for tracking the development of embryos, while genetic screening detects chromosomal abnormalities or genetic disorders. Embryo cryopreservation. Successful embryo culture is not just about the embryo transfer itself. Embryo cryopreservation allows for freezing and storing healthy embryos for future use, avoiding the need for repeated ovarian stimulation cycles, and increasing the chances of successful pregnancy in the future. Laboratory techniques and staff training. Proper training and experience of embryologists in handling embryos, biopsies, and genetic testing are vital for reducing errors and improving success rates. Continuous quality control of the laboratory equipment and conditions is also essential to maintain optimal outcomes. Personalization of ART protocols. ART protocols should be personalized based on the woman's age, ovarian reserve, and specific fertility concerns. Tailoring ovarian stimulation protocols and adjusting embryo culture conditions for individual cases can enhance ART outcomes. Fourth conclusion. Embryo culture and the laboratory environment play a pivotal role in optimizing the success of ART. Carefully controlled conditions, high-quality culture media, advanced monitoring techniques, and skilled embryologists all contribute to improving the chances of successful embryo implantation and pregnancy. Continued advancements in technology and better understanding of embryo biology are expected to further improve ART success rates, making assisted reproduction more accessible and effective for those facing fertility challenges.